All right, well, welcome. I'm Carla Sinopoli. I am Director of the Museum Studies Program here on the U of M campus, and I'd like to welcome you to the first of our fall evening lecture series, which we've entitled Transforming Education, Conversations about the Past, Present, and Future of University Museums. As many of you are no doubt aware, the University of Michigan is celebrating its bicentennial this year, and our series is intended to acknowledge the essential role that museums have played in the university's history. When the university was first imagined in 1817, its founders envisioned museums as essential to the ambitious territory-wide educational system that they sought to create. The university's first museum exhibits opened to the public in 1841, the same year that the first class of students arrived in Ann Arbor. And indeed, as one of our colleagues has recently pointed out, the university had begun accumulating scientific collections even earlier, before there were any students on our campus. Today, 10 University of Michigan museums and multiple galleries house vast and diverse collections, host countless exhibitions, and engage thousands of students in their classrooms, laboratories, and public spaces. So we hope to use this series to both celebrate our museums and collections and think more broadly and critically about the place of museums in the 21st century university. Rather than formal lectures, we structured this series as a set of conversations between University of Michigan museum leaders and distinguished peers from other institutions. And we set our speakers a modest goal: tell us a bit about the past of their institutions, but more importantly, to explore together their visions for the future contributions of their kinds of museums to education, research, and published engagement in the context of the 21st century research university. And I am especially delighted that our lead-off conversation in this series features two zoologists from two foundational and historically closely related university museums. So before I turn the floor over to them, let me briefly introduce them. Jim Hankin is on that side, is curator of herpetology and director of the Harvard Museum of Comparative Zoology, where he's also the Alexander Agassiz Professor of Zoology. After earning his bachelor's and doctorate degrees in zoology from the University of California, Berkeley, he did postdoctoral work at Dalhousie University in Nova Scotia, and then took a faculty position at the University of Colorado, Boulder. Professor Hankin moved to Harvard in 1999, where he's also professor in the Department of Organismic and Evolutionary Biology and a member of the Biological Sciences and Dental Medicine program. Professor Hankin's research focuses on morphology, developmental biology, and systematics, mostly but not exclusively of amphibians. He's a member of the Executive Committee of the Encyclopedia of Life, a recognized teacher, and author of more than 130 scientific publications, and editor of 4 books. In his spare time, he's an accomplished nature and scientific photographer. Our second speaker, Diarmuid O'Fuil, is chair of the Department of Ecology and Evolutionary Biology and former director and current curator of mollusks in the Museum of Zoology here at the University of Michigan. He earned his Bachelor's of Science in Zoology from the National University of Ireland in Galway and a PhD in Biology from the University of Victoria, Canada in 1987. Prior to joining the faculty of the University of Michigan in 1995, Diarmuid held positions at Friday Harbor Laboratories, University of Washington, and Simon Fraser University, and the University of South Carolina. His research explores the evolution, systematics, and biogeography of marine, freshwater, and terrestrial mollusks. And in recent years, he's had the unenviable task of conducting fieldwork in Tahiti, where he and his team are studying endangered Pacific land snails. He's the author of more than 90 scientific publications and has served as president of the American Malacological Society and on the editorial boards of Evolution and Malacologia. Please join me in welcoming Jim and Diarmuid.
you all for coming. Jim and I met for the first time an hour and a half ago, and we sat down and compared notes, and we have a game plan to talk to you a little bit about natural history museums, museums of zoology, the respective origins and brief histories of the MCZ and the UMZ, the challenges we face in the discipline, and the exciting opportunities ahead. So I'm going to kick off with some very preliminary discussion about the nature of objects in natural history museums and how they differ from objects in many other museums that many of you will be familiar with. So here's an outline of the talk. Excuse me.
I think it's getting your colors.
Thank you. First of all, we're missing some text here. The first line is what's different about natural history museums and their objects, and the origins and histories of the MCC and the UMC. Okay, so the role of museums in culture and history are primarily to act as reservoirs of knowledge, often through the case of objects of great cultural significance. And these are sources of scholarship and education. Many of you are familiar with going to art museums, and this is a very famous painting by Vermeer. You may also be familiar with the recent movie based on this painting. But as biologists, our attention is often drawn to something else, to this. Okay, so this is a biological object, a natural object, man-shaped, but it's not man-made. And as biologists, we're interested in where it came from. So the species that generates this pearl is Pinctada maxima. It's pearl oyster. It lives in the Indo-Pacific, typically coral reef habitats. That specimen is a living specimen. It's been modified by humans to make Mabe or blister pearls, similar to the one in the painting by Vermeer. But that in itself is a natural wild organism that's been captured and modified. It represents a lineage of bivalve mollusks that have its distinct identity for a few hundred million years. It grew in nature, feeding on phytoplankton in the coral reef environment, and it has embodied in its exoskeleton here information about that environment. Information about the food it grew in, the temperature it grew in, and also traces of this genome. Okay? So as biologists, when it comes to natural history collections, the objects themselves, they embody the natural environment they come from, and that's where their value resides for us. Although many of them are also aesthetically pleasing, and even more so in this particular context. Okay? So let's talk a little bit about biodiversity. To find that pearl oyster in nature, you'd have to go to a coral reef environment like this. These are some of the most diverse habitats on the planet. Many of them have up to, say, 100,000 species co-occurring, 100,000 interacting species. The basis of the food chain are these corals with symbiotic algae growing in their tissues. And as scientists, we're interested in how nature works. And there's a central concept in science, a specific definition or meaning to the word biodiversity, or biological diversity. And it's very inclusive. It refers to all living things on our planet, all of them. Different plants, animals, and microorganisms, the genetic information they contain, and the ecosystems they form. And as biologists, we know that genetic information they contain is related amongst organisms back to a common ancestor billions of years ago. We're interested in how all this diversity has originated and how it functions. Okay? And when we take specimens from here and put them into our natural history museums to study, we're taking information that's latent, that's embodied in that specimen, about the evolutionary history and the ecology of these organisms. And because they represent a point in time for us, we can use them later on then to infer all kinds of fundamental, basic biological information that we couldn't otherwise have if we didn't have those specimens. Okay? So, I've taken this image from Richard Barnes' poster that he put together for the Objects in Biology series. Let's look at the fundamental issues that biologists want in a natural history specimen. Okay, so right now we're looking at it aesthetically, but really what we want to know is what is it, who collected it, where, and when. Okay, so essentially we know it's a preserved, identified biological specimen, Pitisteri. The collector was Joseph Steer. He collected in Mindanao in the Philippines, and the collection time is 1874. Okay, so knowing that information, we can place that specimen in nature in a specific location at a specific time. And its preserved body is not just its own genome, but all of the material resources it's absorbed through local food webs while it was alive. And that specimen is our portal to that natural ecosystem in the 1870s. Okay? So that's the general background about specimens in biological research collections. And I'd like to turn it over to Jim now, and he'll give us some history of them at CC.
Thank you. Hi, everyone. So one of the things we want to focus on tonight is what museums are up to these days and going forward. But as you can imagine, museums have rich histories and often very interesting things. So we're going to give you a little snippet of information now, just tracing the history of our respective institutions, the museums, and also show some interesting and perhaps unexpected associations between them that are very rich in the past. So, so we're going to go on to origins and history. So let me start with this painting. This is a painting that hangs in the Concord, Massachusetts Public Library, free public library. It's called Philosopher's Camp in the Adirondacks. It was painted in, actually, in the lines just in the front of the wheel. It was painted in 1858 by a man named William James Stillman. And it's significant, thank you. It's significant in a couple of respects. I'll tell you about the people in it in a moment, but it heralds it was actually a camping trip taken by some very famous Bostonians in the mid-19th century. And it reflected interest in the natural world that was really becoming extremely current in those days. As Europeans who came over to North America were migrating west, they were just overwhelmed by all of these novel environments and creatures they were finding. And there was basically an environmental movement that was born in the latter half of the 19th century. And this painting actually helped contribute to it. Now, what's significant for me is some of the people here who are here. This is a very famous, larger-than-life figure in the late 19th century. That's Louis Agassiz. He's the founder of my museum. And Agassiz was a renowned ichthyologist, and he's got a fish on the stump right here, and he's showing the anatomy of a fish to other people. This is Ralph Waldo Emerson, Henry David Thoreau. These were all the transcendentalist thinkers of New England at the time, and they were featured in this painting. Now, then I bring this up because a lot of natural history museums in North America were founded in this time of tremendous interest in the natural world. Here's Louis Agassiz, who you just saw in the previous painting. He founded the MCZ. There's a very interesting association between the Agassiz family and Michigan, but it's not due to Louis, but it's due to his son, Alexander Agassiz, who succeeded his father as the director of the MCZ. And the connection comes in by the fact that Alexander Agassiz got a good tip about a potentially valuable copper mine in the Upper Peninsula of Michigan, and he borrowed $10,000, and he invested in this copper mine and became the president of this mine, which generated half the nation's copper in the 1870s and 1880s. And he became a fabulously wealthy man and left much of that wealth to the Museum of Comparative Zoology. I feel silly teaching you a little bit about Michigan geography here. But here it goes. There's a map in the lower portion showing here we are in Detroit and Ann Arbor down there in the lower right. And, of course, there's the Upper Peninsula in the box, the rectangle in the upper left. And then blown up in the larger image is that circle includes the town of Calumet, Michigan, which was founded by Alexander Agassiz. And it's now, as many of you may know, it's now a National Historical Park devoted to showcasing the copper industry in the 19th century. And this was a very wealthy time for the people involved in this. And they have a club in Calumet called the Misquawabat Club. It's also nicknamed the Millionaires Club, at least at the time. Here's a picture, a photograph that hangs in that this is a few years after Alexander died. Alexander Agassiz actually founded this club. You can see the individuals. Many of them are very important to the past in the Michigan economy, shown here. So it attracted a lot of very, very wealthy individuals, some of whom had contacts and communications. with Harvard and MCZ. And here in Calumet, they ultimately, in the 1920s, dedicated a bronze statue by a very famous sculptor in the center in Agassiz Park, it was so called, in Calumet. And that's in 1923. And here's a photograph. We've actually been corresponding with the Park Service, having to do with who actually owns this statue. It's somewhat contested. But this is a photograph of the park as it still stands today. It was taken a year ago. What's interesting is that many people in Calumet, or most people in Calumet, have no idea that Alexander Agassiz had anything to do with Harvard University. And most people at Harvard University, including the MCZ, have no idea that Alexander Agassiz had anything to do with Calumet, Michigan. But there's a very important linkage between the two. Now, going back to Louis, Louis was not a shy man. And here is an interesting quote which embodied what his and other people's sentiments in those times. In the 19th century, North America was not regarded as competitive intellectually with Europe. And so there were attempts in different parts of the United States and elsewhere in North America to raise up our standards of professionalism in academia to compete with Europe. And Agassiz, who was trained in Europe by some of the masters, the finest scientists of his age, was determined that, as he says, my earnest desire is to put our universities on a footing with those of Europe or even ahead of them. So there would be no longer a disposition to send our students over to Europe to receive their doctorates and so forth. So he had very grand ambitions, and these grand ambitions were embodied in how he ran our museum. And I suspect that they also applied to this museum, these museums here. Those of you who have ever visited the MCZ or come to Harvard campus, this building still stands. This is the original museum building. It still stands, although right now it's surrounded by all other, the MCZ has grown in all different directions, and it's literally grown up, and it's grown underground, and so forth. But that original building is still there. And here is one of the rooms that we have that retains the original architectural style of the building is still intact and still maintained that way. And just we're now going to transition, I think, to UMMC.
You had another point to make?
No, I thought there's another illustration coming later.
Great, thank you. That's great. So the origins of the UMMZ, Museum of Zoology collections, date back to a very specific date, 1837, the foundation of the state, and the charge to set off and do a state geological survey. And actually, that state geological survey, I believe, was the first to find evidence of copper deposits in the Upper Peninsula. So there's a neat linkage here. But as part of that charge was the notion that specimens, geological specimens, plants, animals, fossils, would be set aside in the new campus for the University of Michigan at Ann Arbor in the Cabinet of Natural History. Okay? And that was the very first nucleus of our collections. And we still have some of those specimens today. Here's the rough-legged hawk, collected, shot, unfortunately, collected in 1837 by Sager. Ben Winger, who's our ornithologist expert, told me that it must have been shot in winter because that species breeds in the Arctic in the summer. Okay? So we can get some finer temporal resolution. That specimen has been mummified, essentially. Its internal organs have been removed, it's been dried. As long as it's kept dry and out of light, this specimen will remain intact for centuries. Okay? And again, in its body, it has its genome. It's got everything it's eaten. All the isotopes from that environment are embodied in there. And we have that, plus other specimens from that initial collection. So the cabinet eventually took this form. Okay? It was in Mason Hall, if I remember correctly, and was involved in teaching, especially in zoology. You can see some of the birds specimens here. And by 1850, the cabinet had about 5,000, mainly Michigan specimens, geological, zoological, and botanical specimens. Okay. So soon afterwards, the museum took a rather different turn. It looked outward at the world in a very spectacular way. I want to spend a little bit of time on this because it's rather interesting. Because if it had remained a regional collection, we really wouldn't be having this discussion today. Okay? And that was really the result of an extraordinary individual, Joseph Beale Steer, like Agassiz, larger than life, a homegrown person who was really started off in law but really got interested in natural history. He had a cousin who ran the local newspaper, and he worked out a special deal that he would engage in this circumglobal collecting expedition to, first of all, to South America, across the Pacific to the Philippines, and other parts of Asia, and eventually back to the U.S. over 5 years. Here are some University of Michigan students with him. Slightly different phenotype in the 1870s. Okay? And he was especially interested in collecting birds, but he collected plants, musical instruments, items of anthropological interest. And he would write weekly reports that would be published in the local paper, which created a lot of interest, and there would be regular shipments of specimens back to the U of M. Now, intellectually, what's going on here is the whole world is coming to this campus in what was still a fairly isolated part of the nation in the form of these specimens and these reports from the headwaters of the Amazon in Peru to remote islands in the Philippines, accounts of people, accounts of specimens, adventures, all of this, and material, musical instruments, you name it. Okay? And such was the flood of material that it encouraged the development of a standalone museum building. Now, this may not look familiar. It no longer exists. It was in State Street, served as a main museum for about 40 years or so. This housed the specimens. Steer was made a curator. He was eventually forced to retire because of religious difficulties. He was conflicted. He was a deeply religious man, and he was conflicted about Darwinism, as was Agassiz, if I remember correctly. And he was also conflicted about prohibition. He didn't like, he was a strict teetotaler. And there was a lot of tension with especially students of German descent, who were rather more open to imbibing ethanol. Okay? And this conflicted with his ability to be an educator, and eventually the Board of Regents forced him to retire, although he lived a very long time and kept an association with the program. The next...
Actually, if I could just interject.
Of course.
Agassiz was actually, that's not true. Louis Agassiz was not conflicted. He was completely confident that Darwin was completely wrong.
Right.
And in fact, he founded our museum basically to gather specimens to disprove this very heretical notion that was coming across the Atlantic.
So he was an energetic and useful failure.
Yeah, absolutely.
Excellent. Well, that's one of the things about science, by the way. It's all about data and evidence, okay? And if we could prove Darwin was wrong, we'd throw him out immediately. But we can't, right? But that's very typical for 19th century biologists in particular, right? That tension. And we still see some of it in our classrooms today. Okay, so the next bigger, larger-than-life figure is Alexander Ruthven. The Ruthven building is named after him. He went on to serve as president of U of M for quite a period. He was a herpetologist and was determined to promote museums and museum collections, bring them to the forefront of science and education. An extraordinarily energetic person, highly influential, very proactive. Was appointed the head of the first director of Museum of Zoology, which was given an independent status, I think in 1913, and successfully gathered influence both at the university and at the state level to build a brand new museum complex that opened, I believe, in 1928. And Carl, I understand that one of his inspirations was actually the MCZ in designing the structure. Okay. And right now we are in the midst of transitioning from there to a new research museum center on Varsity Drive. We'll be there next year. It will house the collections of not just zoology, but also paleontology, anthropological archaeology, and the herbarium. And the facilities are fantastic. It's a little bit far from Central Campus, so it has some challenges for our program that we will work hard to address. But the university has invested approximately $40 million in this relocation, so it's a major investment into the museum's programs and their longevity on campus. The Public Museum of Natural History, which is in Ruthven, will migrate to our brand new Biological Sciences building, just east of here. And the interesting thing about this is that within the biology building will be that public museum with specimens, including our iconic specimens, many of them which are the direct responsibility of Dan Fisher, especially the very large extinct elephants, and with a restaurant and lots of traffic back and forth, although our main research collections will be 6 miles south of it. Okay, and over to you now, Jen.
We wanted to close this section, just begin talking about bringing it up to the present. And I put in this slide because it's very interesting what's happened here in Michigan over the last several years with respect to your museum, and the fact that there's this series of conversations discussing why do we have university-based museums? What should they be doing? I guess you could say, should we have them? What's the answer to that? Assuming the answer is yes. What should they be doing, and how are they sustained? It's important to recognize that conversation is happening in many, many universities around, at least in the United States. And we've certainly had it at Harvard over the last several years. And one of the manifestations of it is this new entity. We love acronyms at Harvard, and we can't get enough umbrella organizations. So a new umbrella organization was created at Harvard a few years ago called the HMSC, the Harvard Museums of Science and Culture. And this brings together several, as it says down at the bottom, One World, 4 museums. That's actually simplified. It's actually One World, 6 museums, but it gets even more complicated than I have time to tell you about. But this is bringing together, in terms of the public programs of anthropology and the history of scientific collections and Semitic museums and natural history, in order to better serve the campus community and the public at large, and also more effectively integrate collections, the museum's collections, into the university, into education, into research, and so forth. So a lot of universities are questioning right now, why do we have museums? Given that we have museums, what can we do to make them be better, not only better serve other parts of the university, but function better themselves? Now, we're going to move on to the last 2 sections that we wanted to discuss are what we're calling here Relevance 1 and Relevance 2, having to do with just a couple of examples of why museums are so particularly relevant and important today. I think you're going to do this. Okay, thank you.
So the biologists in the audience don't need to be told this, but here are some general reasons why natural history museums are important. They're really tied in with our success as a species. So, unfortunately, right now, present-day extinction rates are much higher than background rates, and approaching that of mass extinction events. And the biggest driver is our explosive growth in human populations. This little dip is a plague in medieval Europe. This number is out of date by about 20 years. If you get this link, it's rather sobering just to just watch these numbers click over. Okay, let's have a look at that. So that plot I showed you is 20 years old. The number was 6 billion. Now it's over 7.5 billion. Right. And of course, the reason for that is modernity. Is the Industrial Revolution. It's better health. It's better education. But the outcome of that on the rest of the planet is quite severe. Okay. And we really don't know at what number this will level off at. It could be 9 billion, it could be 11 billion. But that number will make a huge difference, especially as more and more people undergo modernization and attain a higher standard of living, seeing most recently in China, where hundreds of millions of people have moved out of poverty. But their impact on the environment then scales up yet again. Okay, so this is probably the issue. Okay, and the impact in terms of extinction rate is huge. Just some examples. These are historical examples of extinctions, and I'm using birds here because they're familiar and we have a lot of empathy to them. The historical pasture pigeon, that was by far the most numerous vertebrate in North America until we killed every single one of them, essentially. We have specimens in our collections with eggs and nests. Many places around Michigan are named after them. Okay. Somewhat more recently, Ivory-billed woodpeckers. We've got a beautiful drawing here by Audubon, last seen in the 1940s. Again, due to deforestation, we destroyed their habitats. A little bit more enigmatically, North America's only native parrot, Carolina parakeet, which went extinct about a century ago. Again, with a drawing by Audubon. This one is looking right at you. Okay, so where are we? About half of the surface of the planet is being modified for human use. Okay, so pastoral, agriculture, urban. Much of the other half is either desert or tundra. Okay, and that percentage keeps on going up. Over half of the world's freshwater, we capture it and modify it for our use. So that foretaste of extinction with those highly charismatic birds is just a foretaste of a wave of massive extinction that will happen this century. Okay. How do we know what we're losing? We need inventories, we need up-to-date knowledge, and we need historical perspective. And natural history collections provide all that. They're some of our best sources of information about fundamental biodiversity and changes in that diversity over time. The other big driver that really hasn't kicked in yet in a serious way, but it scares many, many people, is climate change. Okay? So this is a very famous body of scientific data. It's a Mauna Loa atmospheric CO2 rate in parts per million. It's the most verified piece of data in science. It's verified constantly. You can go to this website and you can check it out on a daily basis, on a minute-by-minute basis. And it was started to be collected in the late 50s and 58. And you can see the seasonal oscillations here, but the very rapid ratcheting up after the Second World War of CO2 emissions. Okay. And the earlier estimates are from bubbles trapped in ice cores. This is the pre-industrial number. We're set to double that. Okay? And by doubling it, we're talking about a radically changed climate and a radically changed planet. Perhaps up to 3, 4, maybe even 5 degrees Celsius higher. And this is this is happening right now, and it's happening very quickly. And a lot of it will kick in by the end of the century. Okay? We can see its impact in a relatively benign way in terms of species distributions in museum collections. Here's a picture of my colleague Phil Myers, who's a mammologist, an emeritus curator of mammalogy. And for many years he's looked at the distribution of small mammals in the upper and lower peninsula of Michigan. He's got some data here on the composition of mammals in the northern parts of Michigan's Lower Peninsula from 1875 to 1980 based on museum collections. Roughly half and half between these two most southern species, the more northerly ones, and since then very rapid change with the southern species taking over. It's happening in real time, and again, museum collections are our best example of that. Okay, so those are the challenges. Massive loss of natural environments, incipient mass extinctions, and very rapid changes in climate and in ranges. Okay, so in terms of our ability to respond to that, one of the really interesting things is right now, at this point in time, once you have these collections, and again, the older they are, the better they are, because they represent populations and sometimes entire ecosystems that no longer exist, and give you that historical depth where you can actually recognize change as it's happening and project it. We have amazing new breakthroughs in terms of our ability to get information from those specimens and put it to use. Okay? And a lot of it is tied in with an explosion in technology associated with getting genomic data and being able to extract that from using specimens. And also our ability to handle and communicate vast amounts of actual metadata through digital revolutions from collections. I'm going to touch on the genomic stuff very briefly, and Jim will take you through the digital revolution and what we can do with it. So this is a rather famous graph showing the decrease in cost of sequencing genomes since the turn of the century, okay, up till 2 years ago. And essentially, at this point in time, it would cost about $100 million to sequence a genome. Okay? When the first human genome was sequenced, it was a huge thing because of the sheer amount of effort involved. Okay? This is Moore's law, which is the classic law about the ability to increase memory on computer chips with time. This is much more rapid. Right now, we're talking about almost $1,000 per genome. Okay? Now, the good news about this also is that the nature of the genomic information we need to access that information has gotten much more plastic, in that we can take even degraded genomes and sequence small chunks of DNA and stitch them together using computers. And one of my favorite examples of this is from a few years ago, from a tiny bone fragment, a finger fragment from a cave in Siberia, of an extinct lineage of human, a relative of Neanderthals. So this is a replica of the finger bone that was excavated. And from this, the entire Denisovan genome was generated. And based on that specimen. Now, again, having the material is a critical thing. Without the material, a photograph won't do. You can't extract the information. Okay? So the promise of these new approaches, when we have these world-class collections of millions of specimens and what you could do with them, the mind boggles as to what higher level resolution we can get of new insights into evolution relationships. Okay, Jim. I recognize that guy.
Yeah, so we're going to go to a brief graphical moment here. So I just want to follow on what Dharma just said. So I mentioned a few minutes ago that the people have questioned, what is the role of museums today? Well, it turns out that we now recognize, very broadly, society at large, I think, recognizes that museums have, their collections represent, they're gold mines in terms of the kinds of information and resources that they represent. But also, at the same time this was represented, is one of the problems was immediately recognized. There is a serious issue with gaining access to that information and gaining, whether it's data written on a museum tag that's sitting in a jar of ethanol, or a tag that's tied on the edge of a specimen, or something about the skeleton inside. And so both within the United States and even internationally, beginning, 20 or 30 years ago, in combination with the growth of digital technology, which you're all familiar with in one way or another, there's been a tremendous push to convert museum data and resource into digital form. And I'm going to trace that in my own personal history in a couple of slides. Picture yourself in 1974 as a graduate student, and your major professor tells you in your discussions what you're going to do your dissertation on. Your major professor suggests you work on a particular genus of salamanders that's found only in Mexico, which you barely ever heard of. And your assignment is to find out where you're going to go down and collect some, but you have to find out where to go. Well, one of the ways you do this is you contact other museums, and you say, well, what specimens do you have, and where are they from? So this is what I did. In those days, this is before email. This is hard to imagine for some of you. This is before the internet, before email. You would write a letter. Now, a letter, for those of you who don't know, you take a piece of paper and you write it and you put it in an envelope. You send this letter off to different museums and institutions, and you introduce yourself and you say, can you send me information about what you have? And you do that, and then a few weeks later, you get this kind of information. The museum curatorial associate or curatorial assistant would have pulled out their index cards and put the index cards in a plotting of a Xerox machine and photocopied the index cards, folded them up, and sent them back to you in a letter. And that's an example of one such. I can't remember if this is from the Smithsonian. This might have been from the University of Michigan, for all I know. I can't remember which, because you have some of these things here. That was the state of the art in 1974. Okay, let's fast forward to 2017. You can go to a website for vertebrate organisms. You go to VertNet, which has amassed data records for vertebrate collections, many of them in North America and elsewhere. And you type in the word Thorius up here, which is the name of the genus, and you hit go, and within a second or two, you find out that it's displaying for you allowing you to access 8,843 records. And here are just 3 of them, the first 3. And just by coincidence, these 3 are in my institution, but there are many, many more, of course. And then you can then click on, you can actually then go to the iDigBio website. I'll tell you more about iDigBio in a moment. And you can pull up those records of Thorius. And not only that, but you can instantly generate them if those records have been georeferenced. In other words, if there's a latitude and longitude associated with the locality. You get a nice map of showing you where those specimens are found. And it's a great way to find out if there are errors in the records. This is an endemic Mexican genus, so the fact that there's a lot of dots in Mexico is to be expected. What's unexpected are 2 dots over here in Africa. What are Thorius doing in Africa? And you can then go on to click over those specimens and, and find that actually the longitude, the plus and the minus on one of the records has been reversed by whoever typed it in. Fortunately, these are not the Michigan records and they're not MC's. In order to protect the innocent, I won't name which institution's records these are. You can still find it out today. But it's a great way to check data aspects of your records, but also to get tremendous information in a short amount of time. Now, how did this all come about? Well, for vertebrates within the United States, there have been a series of initiatives, and just going back to, I don't know if you can read the dates here, back to 1998 all the way up to 2012, there's been a series of NSF grants that have been paying to digitize collections and put these data online. And then there was another one called VertNet, yet another NSF grant that was not taxon specific. Some were originally fish for mammals, birds, and so forth. There's VertNet. And then the real game changer that we're in the midst of right now is something called the ADBC program, sponsored by the National Science Foundation. And that the ADBC program, here is the acronym means, we're in year 6 of a 10-year program by the National Science Foundation, and the U.S. is spending $100 million to digitize museum collections. Now, on the one hand, by any standard, $100 million is a lot of money. But actually, by the standards of how much is it going to take to do these collections completely, that's not nearly enough. But it's making more than a start. And you can see as of June 8th, it's really been a successful program. And all of those dots on the map shows all of the collections that are participating in the ADBC program. So it's been a tremendous success. But I just want to emphasize also, this is not just the U.S. We're not even leading the effort here. There are some other countries that are actually way ahead of the United States in both technology and the comprehensiveness of their approaches. Here's just a list of several initiatives and the countries that they're associated with. And what's also pretty cool is that data from all of these countries, including the United States, are aggregated on a global scale into one entity called the Global Biodiversity Information Facility, which is based in Copenhagen, Denmark. And here is the GBIF data. As of last June, there were more than 700 million georeferenced records. And you look on the globe there, the colors represent the density of records. So white is no records, yellow is some records, and the more deep the red gets is the greatest intensity of records. So you can instantly see where there's been a lot of collecting effort and where there hasn't been a lot of collecting effort, or at least where the specimens are not digitized. I suspect there's a lot more records from Russia and what used to be the Soviet bloc, but they just haven't either digitized their records or released them yet. Now, that's impressive, but as I alluded to before, it's a massive task. And there are estimated to be 3 to 4 billion specimens housed in natural collections, which offer a tremendous resource and for all kinds of studies. We estimate that there's less than 50% have been digitized, even in terms of their core data. Many fewer are georeferenced and imaged, and it's the special collections, what I'll call special collections, have largely been touched, but they're beginning to be touched now. What do I mean by special collections or projects? Well, it's this kind of thing. It's using digital imaging technology. Now, this is a spectacular micro-CT computed tomography scan of an animal that, as far as I'm concerned, never attained nearly as much glory in life as it attained. This is an earthworm that was collected on 26 Oxford Street in front of the Museum of Comparative Zoology. It was not a very exotic collecting event. But this was scanned in collaboration with a journal called Giga Science by my Harvard colleague Gonzalo Giribet and people in his lab. And it is showing the detailed internal anatomy of an earthworm, and then it has been each of the different organs and organ systems have been differentially coded. This is actually a much more complicated image, and you can download from the MCZ website a so-called three-dimensional PDF and rotate it in 3 dimensions and pull out all different organ systems, and even some of them even have been labeled. But this just gives you an idea of the kinds of things you can do. It's a kind of non-destructive imaging. You don't hurt these specimens at all. If they're sitting in ethanol, you scan them and put them back in the jar, and they're really just as good as before. But you can get tremendous amounts of information. And these kinds of images are really transforming a lot of disciplines, not only research disciplines, but they make tremendous teaching devices. Now you can generate a on a 3D printer a plastic version of this, and we're doing this for a lot of amphibians now, that make great teaching tools. And this brings me to a project that, a very ambitious project that actually your museum here is part of, my museum, as well as about another 10 institutions across the United States. This is the so-called OVERT TCN. It's a thematic collections network, which is going to image, I think the claimed goal, let's see if we can pull it off, produce micro-CT scans for 10,000 vertebrate species. And here we see, this is a great illustration prepared by the people at the University of Florida, showing you for a single frog, you can make, with a single scan, you can then manipulate the images in various ways to pull out all different organ systems. You can look at the muscular system, you can look at the circulatory system, you can look at the gonads, you can look at the skull and shade it, look at it in different perspectives, the inner ears and so forth. It's a tremendous, tremendous opportunity for all kinds of studies. I think there's a video here. Let me see if I can get this nice video that was produced by Science magazine.
Scientists are embarking on a massive effort to CT scan.
Let me just go back. Here we go.
Scientists are embarking on a massive effort to CT scan all vertebrates, from bats to turtles to squirrels. Countless animal specimens are preserved and stored away in museum collections, making them difficult for scientists and the public to access. But the plan to scan all vertebrates will change that by creating 3D models of the animals and uploading them to a free online database. Over the next 4 years, 16 universities and museums across the United States will scan more than 20,000 fluid-preserved animals, covering about 15,000 different species. Even though that's only 22% of the 68,000 vertebrate species, they will be carefully picked to cover over 80% of living vertebrate genera. To speed things up, researchers have made fish burritos, where multiple specimens, such as these piranhas, are wrapped, stacked, and scanned together. This unique view of vertebrates has already led to discoveries, including new species, such as this claimed fish that was hiding in a museum for several years. They can also help scientists understand new quirks about animal diets and lifestyles, such as this Philippine flat-headed frog that ate a crab before being preserved, or this other frog that's so tiny the ant inside its belly is almost as big as its head. Texas A&M will be in charge of scanning anything large. In the past, they've scanned an eel, a bowhead whale fetus, and even a shark. The project will also pick about 1,000 specimens to soak in an iodide dye, which boosts the contrast on soft tissues, improving the view of the muscles, circulatory system, and brain. Scan All Vertebrates is a basic science initiative to inspire new research, and is a huge leap forward in digitally preserving and sharing biodiversity on Earth.
So that's just one way that I think museums are really kind of leading the various areas of research and teaching, and really just kind of scratching the surface of the kinds of opportunities there.
Great. One or two more things to finish up. And by the way, Cody, I think you're one of our local people in Overt, along with Dan Rybowski and Alison Davis-Rybowski, right? Correct. Okay, good. And it's a very exciting project. Notice how multi-institutional these ventures are. I mean, so the museums really are part of the same team addressing the same questions. And by pooling our resources, we make basic fundamental scientific discovery and educational outreach that much easier. I'd like to finish with one little thing applied for one of our new hires, curator Alison Davis-Rybowski, who's not here. And I want to focus on this for two reasons. It's a biologically interesting system. It's studies made possible using museum collections. And it has a significant field component, because one of the things we really haven't talked about here is the fact that growing research museums have an active field component, which is a very important part of their programs. So this is a drawing that's done by our museum artist, John Megahan. He's a supremely talented scientific illustrator, and he's got 2 species of snakes here. They were sampled, I think, in 1940 in Guatemala. They're in our collections. Superficially, they look like 2 individuals from the same species, but this is an example of Batesian mimicry, where one species is venomous and the other species has evolved a matching pigmentation pattern. We think this confers some protection against snake predators, so by fooling them. And there's a lot of ecological and evolutionary theory tied in to the evolution of mimicry, especially Batesian mimicry in nature. It's highly controversial, and Alison has recently published a masterful study on this. She's got a small video which explains some aspects of this work and some of her field work, and we'd like to finish with this before we get on to questions.
There's great evidence that coral snake mimicry Coral snakes are a group of snakes that are in this a very venomous group of snakes that contain what we consider to be some of the most venomous snakes on Earth. In order to honestly signal their toxicity to their predators, they have evolved with a very bright, what we call aposematic coloration, where you get red and black banding that's very visually striking. And so their mimics are an entirely different group of snakes. The mimics, they've convergently evolved the same coloration, but they appear to be fairly harmless to predators, and that's what we term coral snake mimicry. Even though there's great evidence that coral snake mimicry exists, it seems not to follow some of the theoretical predictions that have been made by other biologists about how mimicry should work, and also some of the observations we've seen in other mimicry systems, like in butterflies. So what we wanted to do next was test why we might be seeing these differences between what we predict and what we observe. And so the place we went to investigate this was the Peruvian Amazon, which happens to be not only the most biodiverse region in the world for lots of different groups, but it's also the place where you can find the greatest number of coral snake species that live in the same place and the greatest number of mimics. So the University of Michigan team consisted of 2 professors, 1 postdoc, and 4 graduate students. Together, we basically all went and tried to catch as many reptiles and amphibians as we could, because we had some members of the team who were answering questions about the drivers of biodiversity. We had some people who were interested in the ecology of frog diets, or a number of different things. And so we all worked together to collect as much data as possible for everybody who was on the trip. I was there studying coral snake mimicry, and so we worked day and night to try to catch as many snakes as possible by going to Peru and looking at a place that has the highest diversity of coral snakes in the world. You can actually ask questions about how mimicry is operating when there are many models and many mimics. And so I think that until you study those hyper-diverse regions, you won't ever really understand coral snake mimicry.
Okay, so that finishes the presentation part. We certainly have time to address any questions you may have.
I guess I'll ask you one about, thank you, first of all, for a great presentation. I want to ask you a little bit more to talk a little bit more about the university side of your work. Is there anything you think that makes your museums being situated in a university different, say, than the Field Museum or the American Museum of Natural History, who's also collecting large collections and doing scientific research?
Absolutely. I mean, there is different imperatives at university-based museums. I'll give you just a couple of ways. One is obviously the educational component is very strong. And I'll just give you, in my own experience, we have many, just like you do here, we have many university-based museums at Harvard. And I've noticed, I've been director for 15 years, I think, by now. And I've noticed that some of them do better than others. I won't identify which ones. I would say the MCZ does pretty well. But some do better than others, and you can explain it very easily, because we are within a Faculty of Arts and Sciences, I suspect just as you are here. The Faculty of Arts and Sciences has 2 principal priorities. One is faculty research, to be sure, but also is teaching undergraduates and graduate students. And I've often felt if my institution can align its activities to fall under one or both, hopefully both of those priorities, we will have a better chance of succeeding than if we don't. And that's, I look at some of the other museums at my university who I don't think are doing well. That's because sometimes they're not satisfying either of those imperatives. And those imperatives differ from a freestanding museum such as the American Museum of Natural History or the Field Museum, or for that matter, it differs from the Smithsonian, the federal government museum. So that's a pretty obvious way. It's important and different.
I agree with that. I'll just add one or 2 points. One of the intriguing things about being a university museum is your long-term influence, because you train the future curators of other museums. And museums are all about transgenerational information, because the collections are transgenerational. You only have them in your care for a while, and then you pass them off. And so that long legacy of education and training has a special role to play. And I think university museums that retain an active focus on scholarship have that influence. Where it can get a little bit difficult is university museums, our members typically, our faculty members, have their tenure very often in another unit, a non-museum unit. And there sometimes can be some tensions, especially early on in people's careers when they're assistant professors, and there's a whole series of things they must do to become tenured and be successful, between triaging and prioritizing what the tenure home department may think is most important versus what they themselves feel is their responsibility as an assistant curator. That can occasionally be a problem. Some models that seem to work quite well are university museums where tenure is granted within the museum unit itself, because that allows them to define their mission very explicitly and to be more consistent with time. But I think being a museum, and Dan, feel free to chime in here also, being a museum in LSA is really quite helpful. Because I think there's a wider appreciation for scholarship than we would get potentially if our college was, strictly speaking, a science college only. And there are interesting cross currents that come into play that we would not get otherwise.
Could I just give a brief addition? Just say the impact that we have as university-based museums, particularly on undergraduates, is something that I think freestanding museums can't touch. Some of our students who take our specimen-based courses and especially participate in our field trips, they are life-changing. It's not an exaggeration to say they can be life-changing experiences for these students. And they enrich the museum as well as we are able to enrich them.
I thought it was a great presentation. Thank you. I was wondering, so with digitization, at least in our museums, we've seen a lot of cross-communication between the various units that used to stand in isolation, like the Herbarium and the Museum of Zoology. But I think also we're finding out there's a lot shared between our natural history museums and also, like we say, archaeology, biology, and paleontology. Are you finding that in Harvard there are also sort of administrative boundaries that are falling apart? Are curators talking more to one another and sort of jumping across taxonomic boundaries?
Well, yes and no. That's a complex issue. I can get it in a couple of ways. Even within the MCZ, we have certain. When I first became director, we started digitizing. Different departments were using completely different systems. And still to this day, there's one exception that hasn't bought into the program. I think actually digitization at a museum or an institution like ours is as much sociological as it is technological, getting people to buy into common systems. Now remember, my institution is famous as each tub is on its own bottom. So each institution likes to do things their own way. There is the beginnings of a push among the museums to actually link up not to aggregate into a shared database. That would be too revolutionary. but to at least come up with a common search portal to be able to look, let's say, for instance, just take the word Agassiz or the name Agassiz. Alexander Agassiz has material in all of the different collections. There is no way yet to say if you wanted to look up Agassiz and find microscopes that he used and specimens that he called plant specimens, you couldn't do a single search. But so there's now talk about coming up with a single search portal that would allow you to search on any topic throughout the museums. Yale has just done this with the library, and it's a spectacular system. Now that will probably motivate Harvard to change its ways, because something that Yale has done that's better than us is not something that we can tolerate.
We're working on that too, so we may beat you to it.